I propose to address the following question. Does biology make sense without Darwin? The answer is yes, and that's pretty much the end of my talk. We can open for questions. I call it the evolutionary paradigm. The paradigm is everything that's permissible to believe. It's like a box. Everything inside the box is the whole of reality. Everyone in the box is quite sure there's nothing outside the box. That's a paradigm. Thinking outside of this box is just simply unthinkable. Anyone thinking of an idea outside this box is not simply wrong. Their views are unthinkable. And there is an evolutionary paradigm, and it goes pretty much like this. I think Julian Huxley spelled it out as well as any have. Our present knowledge indeed forces us to the view that the whole of reality is evolution, a single process of self-transformation. Now, when somebody believes that the whole of reality is evolution, that there is nothing else, that everything is just spontaneously self-transforming through natural processes, their best argument for evolution is two words. How else? There isn't any other way. They cannot think of a possibility that would not include just random chance processes. And because of this, we have what is called Dobzhansky's dictum. Nothing in biology makes sense except in the light of evolution. If people really believed this, you couldn't obviously allow a person who did not accept evolution to be a school teacher in any of the sciences. They couldn't be allowed to participate in the allied health or medicine. They would be closed from careers having anything to do with science. And sadly, there are people that believe that something like this is true, that nothing in biology makes sense except in the light of evolution. Now, I could point to my own career and say, well, spent about 40 years having a brush with science. I had a double major in chemistry and biology in Minnesota way back in 1900. Got a PhD in cell biology from Brown, and spent a couple of summers Woods Hole Marine Biology Laboratory working on invertebrates, the sea cucumber Thyone briareus, that's a heartthrob for you, and a couple of years in research at Mayo Clinic in Rochester, Minnesota, and 34 years in the faculty of Washington University School of Medicine. And my students said I made sense of biology. In fact, I received a number of awards for teaching. And so I take issue with Dobzhansky's dictum, but you might say, well, you know, one person and you're a creationist, what would you know? Some evolutionists have come to the same conclusion that evolution really is non-essential, and that was my discovery. In the years that I taught at the medical school, I didn't bother to teach anything about evolution. I mean, at one time or another, I guess I've given a lecture on every organ system in the body with the exception of the pineal. I could never get pumped on the pineal. And I could have saved the last 5 minutes of the lecture, say a lecture on the kidney, to speculate on how we come to have kidneys before we had kidneys. But if I would have done that, I would have added nothing to my students' knowledge of renal physiology or anatomy. I would have contributed nothing to their ability to practice medicine. In short, it would have been a waste of their time. I never did it. And in 34 years in the faculty, I never had a single student ever complain. No one ever said, you know, we think Dr. Menton gives a decent lecture. It's just that we feel cheated. He never gives us any evolution. Here's an evolutionist commenting on evolution, and I couldn't agree more with this gentleman. He said that while the great majority of biologists would probably agree with Theodosius Dobzhansky's dictum that nothing in biology makes sense except in the light of evolution, most can conduct their work quite happily without particular reference to evolutionary ideas. That's an interesting admission from an evolutionist. He went on to say, and I quote, "Evolution would appear to be the indispensable unifying idea and at the same time a highly superfluous one." I couldn't agree with this person more. Evolution is obviously important to the person whose whole worldview is based on evolution, that everything has come into being by a process of evolution. I'm sure it's important to them in their minds. But at the level of empirical science, it contributes nothing. I found it possible to not only teach with, but publish papers with people who simply were evolutionists, for the most part, I suspect, agnostics, atheists. And I had no problem interacting, no altercations. Now, if we would have published a paper dealing with how did we come to have livers or kidneys or placentas or whatever, we'd have been in trouble. But as long as we stuck to empirical science, there wasn't a problem. You know, you might think, well, this is the only evolutionist that's ever raised the issue that evolution just might not be all that important. It could be superfluous. But just recently, in an article published in the Boston Globe, October 23 of 2005, Mark Kirshner, who is chairman of systems biology at Harvard Medical School and a very distinguished, well-known evolutionist, Dr. Kirshner said, "In fact, over the last 100 years, almost all of biology has proceeded independent of evolution, except evolutionary biology itself." I mean, if you're going to study evolutionary biology, you need evolution. I hope we're not going too fast, are we? No, we think so. He went on to say, speaking of his own field, "Molecular biology, biochemistry, and physiology have not taken evolution into account at all." Now, to be fair to Dr. Kirshner, he went on then to say that we're going to have to start taking it into account. He wasn't proposing that we ignore it. As a professional evolutionist, he wouldn't. But isn't it interesting? Over the last 100 years, we've had the most incredible breakthroughs in all areas of biology. Think of just molecular genetics, DNA, all of this by itself. And in many other fields of molecular biology and biochemistry and physiology. It has flourished over the last 100 years, and Dr. Kirshner tells us that it really didn't take evolution into account at all. It's useless. It's superfluous. What kinds of questions have they answered with evolution? For example, what about the origin of life? If you can't explain the origin of life, you might as well forget about everything else that follows. Dr. Paul Davies is a physicist. He's the director of the Astrobiological Research Center at Arizona State University. And he's very, very frank in his assessment of what is known. He said, and I quote, "How did stupid atoms spontaneously write their own software, and where did that very peculiar form of information needed to get the first living cell up and running come from? Nobody knows." And I would say you could add "nobody knows" to just about every non-trivial macroevolutionary claim that there is. Where indeed does new information come from? You know, Darwin wasn't even aware that there were such things as information in the cell. Well, the only real source of new genetic information is mutation. You're looking at mutant human cervical carcinoma cells here. They're called HeLa cells. They keep dividing and keep dividing. The person that they were taken from is long since deceased. As far as we know, these cells will keep dividing forever. Normally, cells only divide 15, 20 times, and that's it. It may be part of why we only live to be just so old. But in cancer, one of the things that can change is the cells can continue to divide, but of course they're highly abnormal. At this point, you wouldn't get a cervix out of this, and you certainly wouldn't get a human being out of these cells. But it's mutations that are really the source, if you are to have a source, of fundamentally new genetic information, not just shuffling the deck, but bringing in new genes. And what do we know about mutations? Well, Dr. James Crow, who's chairman of genetics at the University of Wisconsin Medical School, knows a bit about genetics, I suspect. And he says that the typical mutation, thank God, is very mild. It usually has no effect, we should say perhaps no obvious effect, but shows up as a small decrease in viability or fertility. Now, this is interesting because natural selection, which we hear as being the explanation for everything, it's not chance, the evolutionists will tell you, it's natural selection. Takes everything out of the realm of chance, makes it inevitable. I wonder how many people here could define natural selection. Particularly if I were to say, I'll allow you 2 words. Natural selection can be defined in 2 words. It's called differential reproduction. Basically, do you leave more offspring or don't you? It has nothing to do with whether you're the biggest, meanest animal in the forest. I mean, you go around kicking all the other animals and taking their food away. If you don't leave any offspring, what's the difference? It's differential reproduction, simple as that. And yet, Dr. Crow tells us that the typical mutation shows up only as a small decrease in viability or fertility, precisely the opposite direction of what we want for natural selection, where we want to have a progressive increase in the number of offspring left. One gentleman who studied this whole business of mutations and natural selection a great deal is Menton. He's an evolutionary geneticist of some renown. And Chimera came up with what's called Chimera's distribution. Shows the effects of mutations on a population. Now, look at Chimera's distribution. The first interesting thing is his curve in red doesn't even come up to neutral because he says he doesn't know of any truly neutral mutations. So he stops his curve short of neutral. You may want to check out the curve on the right side, the beneficial mutations. He doesn't know of any beneficial mutations for organisms living in their normal environment, in other words, apart from exposure to drugs and what have you. As we tail off to the left, we notice they become lethal. Now, here's the thing that's intriguing. Chimera came up with this. He said, we have a no selection zone. You can't select for the suboptimal mutations in the no selection zone because there's not enough effect on the phenotype. There's too many layers between the mutation in the DNA and the final phenotype and its effect on leaving offspring. Most mutations just fall below the radar. And you know what this means? It means we are collecting them. It's called a genetic load. In every day and every way, that genetic load increases. You know, if there were some beneficial mutations, and I just threw this one in on the right side. I mean, if there were any, Chimera didn't put any in, but if there were, they would be small in number. And of course, they would not be immensely, outrageously, uproariously positive mutations. They would very likely fall in the no selection zone. Well, I wanted to share with you some particular things that I've thought about, some things that I've worked with, published on over the years, and to look at them from the perspective of, did evolution have the answer? Or did the creationists maybe have a better read on things? Let's start with the human eye. Got a question for you. Human eye, bad design or bad science? Because some people say the human eye is very badly designed. Give you an example. Frank Zindler, a retired biologist, says, and I quote, "Although the human eye would be a scandal if it were the result of divine deliberation, a plausible evolutionary explanation of its absurd construction can be obtained quite easily." You see what he's saying? He's saying that the eye is so badly designed that if a god of any kind were to lay any claim to it, it would be a scandal. And look what he goes on to say. However, you expect junk from evolution. After all, you take a process that's mindless, purposeless, goalless, random chance processes. Try going on a fishing trip sometime that way. With no planning, no goal, no objective. And so he's saying that if a God were involved, it looks really very poorly put together, but you expect poorly put together things from evolution. That's why he says a plausible evolutionary explanation of its absurd construction can be obtained quite easily. So the eye is just simply absurd. Why does he say that? In fact, a lot of biology textbooks make this very point. This is why. You're looking at a section through the retina. Took some years ago with a light microscope. And it's the full thickness of the retina. What you're seeing there is actually thinner than a sheet of saran wrap. It's that light-sensitive layer in the back part of our eye. And it's part of the brain. Did you know that? Really, the whole eye, with the exception of the cornea and the lens, is part of the brain. You young people, when you begin to date, you might want to check out the eyes of your future spouse, and you can check out their brains before you get married. Because it's part of the brain, you do not have nerves in there. They're fiber tracts, and you have ganglion cells and what have you. And so you see the layers. It's like a layer cake. On the more superficial aspect, we have the optic nerve fibers, and then we have optic ganglia. We have bipolar neurons. There are other kinds of cells and nuclei in there. And then way down on the bottom, that's where the photoreceptor cells are. These are those cells that you may have heard of that have the rods and the cones. Cones are sensitive to colored light and rods to just black and white. And then at the very bottom, you get the pigment layer. This is a layer of the retina that's just full of pigment. It's pitch black down there, and it has a virtual lake of blood. Not just blood vessels, just big, huge channels full of blood. And the reason for that is these photoreceptors at the bottom are among the most highly metabolically active cells in our body. They require a lake of blood. And what the evolutionist is saying is this: where's your God? He got the film in the camera upside down. Years ago, when I first took photography, we shot with these big 4x5 sheet film cameras. Boy, am I revealing my age or what. And we had to load the film on a film holder. And you did it in the dark. If you weren't careful, you got it in backwards. And you get that old heavy Estar-based film we use, you get that in backwards, you take a picture and you develop it, you get nothing. We used to call them ukulele pics at that point. So the evolutionist says, your God got the film and the camera upside down. You see the light's coming from the top down, as you see there. And at the very bottom, those little dots are the rods and cones, or the distal ends of the photoreceptor cells. So a photon of light, if you will, has to come down from the top, as that arrow did, going through all those layers. You can imagine bouncing off all these fibers and all of these nuclei and just kind of bouncing all over the place until it finally gets down to the bottom. And the evolutionist proposes that, well, there goes your resolution. The retina is going to have really lousy resolution because the photons have been bumped around through all that stuff. And then, of course, it's going to interfere with light sensitivity. Well, we have two problems just for starters. What they say is certainly true enough. It would appear that it would interfere with resolution. It would appear that it would interfere with light sensitivity. And yet, neurophysiologists have demonstrated that those rods and cones are sensitive to single photons of light. And as far as resolution is concerned, the retina is better than the lens and the cornea. Our eyes are what we call refraction limited. They're limited by the optics, not by the retina. The retina is way ahead of the optics. And so it would appear, although we ought to take a hit given the way the retina is put together, we don't. There are very good reasons why you want the retina upside down. You want to keep these very demanding cells right down in that lake of blood. Another reason is when a photon comes down through that retina, when it finally gets to the bottom, it fires the photoreceptor cell. And then the next thing that must happen to it is it has to be trapped in that pigment layer, the retinal pigment epithelium. Because if it reflected off things and then came back and hit the receptor a second time on a bounce, you'd burn out the receptors faster than you do now. And you have any idea how fast you're burning out your rods and cones right now? They have to be turned over about every 2 weeks. So there are good reasons for having it down there, and I could give you a lot of others. But the evolutionist says if there were a God, he would have had the retina facing the other way with the photoreceptors pointing towards the light. In fact, they point out that there is one group of animals that have a proper retina, laid out as an evolutionist says it should be. And that would be the octopus and what have you, the cephalopods. You know, every vertebrate, every creature with bones has an upside down retina like this. And one of those bony creatures happens to be an eagle. And I used to go down and watch the eagles along the Illinois River when they came through, and I was living in St. Louis. And I attended a class over at Pierre Marquette State Park, and the naturalist said, you know, an eagle can sit up at the top of a tree, look down the river and see a fish just under the surface of the water up to a mile away. You know, just once, I'd like to have an octopus. Take the octopus down to the river, lift him up into the tree, aim him downstream and say, check it out. But let's be fair, I'll bet that octopus sees better where he lives underwater than the eagle would if you put him down there. So a special eye for each. Well, you know, an evolutionist looks at this. Would an evolutionist even study why the retina is upside down? No, because you already know it's junk. You expect junk from evolution, so you ignore it. You say, well, it's upside down. Where's your God? You expect this kind of stuff from evolution. But you know, if you ignore evolution, and I recommend it, if you ignore evolution, as most working scientists do, who work in the field of empirical science, and you go ahead and ask the question, well, you know, it makes sense to have it this way, but why don't we take a hit? Well, just a couple years ago in the Proceedings of the National Academy of Science, not an easy journal to get published in, there was an article that was published dealing with a cell that I used to talk about once in a while when I gave lectures on the eye. It's called the Mueller cell. We've known about the Mueller cell for 200 years. And we just have had no idea what it does. It looks like a tree trunk. It goes through the whole thickness of the retina from the top to the bottom. Everything else is stratified. But the Mueller cell goes top to bottom. Kind of goes down like this through the retina. And this is what they discovered. The Mueller cells are living fiber optics. They actually demonstrate, you see on the left, actual Namarsky photograph of a Mueller cell going top to bottom from the vitreous at the top to the retinal pigment epithelium at the bottom. And you can see it actually conducting light in the right frame. You know what a fiber optic is. Light goes through like a plastic tube. It goes through at 186,000 miles a second. I mean, it doesn't get slowed up as it goes through this tube. And the tube can wander all over the place. Doesn't make any difference as long as one end is lined up with the other end. In the middle, it can do this. And that's what happens, apparently. The Mueller cells are living fiber optics. But imagine if we would have thought as an evolutionist and said, well, we know why it's the retina is upside down. It's because there's no God, and you expect junk from evolution. I'll stick with Scripture. Proverbs 20, verse 12. The hearing ear and the seeing eye, the Lord hath made even both of them. And then there's the famous tailbone. Anyone here ever fall on their tailbone? It's been said that when you fall on your tailbone, there's three positions you cannot tolerate: standing, sitting, and lying down. Those three. And why is that so? You know, the evolutionists consider the tailbone to be a vestigial tail left over from our tailed evolutionary ancestors. Now, let's analyze this thing with a little bit of logic. The vertebral column is a linear structure, right? It's not circular, it's linear. You have two ends. At one end is a head, okay? Now, think about this. The other end has to end someplace. Does it have a choice? No, it must end someplace. And wherever it ends, there will be an evolutionist standing and pointing and saying, aha, a tail. As you know, the coccyx, which means chicken comb, I prefer that name, it's a scientific one. The coccyx, which you see here on the right, is a fused set of about 3 or 4 vertebrae. And it has been described in biology textbook after biology textbook and in popular news articles and on websites as being a useless vestige of our tailed ancestors. I can't think of anything that's further from the truth than that statement. In fact, if you were to describe how useful a bone is in terms of how many muscles attached to it coming from how many different directions, I think the coccyx would win. For example, let's look at it from the bottom now. From the top. We have 3 pairs of muscles that attach to that little piece of bony real estate right there. That's the coccyx. Coming from the front, the pubic part, are a pair of muscles called the pubococcygeus muscles. That tells you the muscle goes from the pubic bone to the coccyx. There is another pair of fan-shaped muscles that attach from the iliac blades, those are the hip bones out here, to the coccyx, and they're called the iliococcygeus muscles. And then there's a third pair coming in, they're simply called the coccygeus muscles. Those 6 muscles form a muscular floor that gives us a floor to our pelvis. Sitting on that floor is the bladder, the uterus, where appropriate, sigmoid colon, such structures as that. Now, since we stand vertically, it's good we have that muscular bowl down here. Because if we didn't have that muscular bowl, the first time you went like that, whoopee. I see, you're getting the point. That's the main thing. Yeah. So it's not useful. But the interesting thing, the evolutionists have changed the definition of vestigial organs. It used to be they were useless, and then once they found a use for just about every organ in the body that was once considered vestigial, then they changed the definition to, well, not as useful as it used to be. And when that didn't really seem to work real well, they changed it once again, and now the definition includes the following. Organs that could be very, very useful, but they're doing a different job today than they used to do. So you see, now the burden of proof is removed completely. The evolutionists can look at the coccyx and say, okay, so it's an important anchoring point for the whole pelvic diaphragm. But that's not what it used to do. We used to swing from tree to tree with that puppy when it was a tail. And then bird feathers. Some of you have heard me talk about bird feathers before, but this is something where I just throw out the challenge to any evolutionist. They believe, of course, very sincerely and deeply that birds have evolved from reptiles. And we won't get into any big details there, except they're convinced that they evolved from dinosaurs. Not all evolutionists, but most. Specifically the theropod dinosaurs like the T-Rex. And you look at a T-Rex, you look at any of these theropods, they get the little short upper limbs. They don't even reach the mouth. And these great big massive lower limbs. We're not even sure what they do with these upper limbs. We know they don't walk on them, they're too tiny. They don't reach the mouth, so they really can't feed themselves with them. They don't seem to be adequate to grab prey. But what do we know for sure they didn't do with them? Now, evolutionists often substitute artistic renderings for data. They hire an artist, and the first thing they say to the artist is, "Would you like to be paid?" Draw me a scale turning into a feather. So here in Nature, very famous journal, back in 2001 was the whole sequence by which scales turn into feathers. They start with an elongated scale, and they say that that scale became frayed, as you see in step 2. And then through a wonderful Lamarckian mechanism, This acquired characteristic was passed on to its offspring genetically. I mean, we know better than that today. What's really interesting is you go from step 2 to step 3A, and you can't go there. Because step 2 is what we call primary branching from one point. Step 3 is compound branching. You don't derive compound branching from primary branching. But anyway, if you're going to be critical, you don't want to go in the field at all. Then, once you get into 3A, you could conceivably go to 3B with another order of branching, which would be somewhat like a feather. Now, what's interesting is how many evolutionists have said that feathers and scales are just dead ringers for one another? Hardly tell them apart. Alfred Romer, a very distinguished vertebrate anatomist biologist, in his book Man and the Vertebrates, said although these structures, that is feathers, seem quite different from the horny scales that cover a reptile's body, the difference in reality is not very great. You probably thought there was a lot of difference, huh? I mean, if Professor Romer tells you there's no difference, you can take it to the bank. And then, I don't know how many of you have the Encarta encyclopedia on your computer. You might want to check it out. Look up feather if you need a laugh or two. And in feather, under feather anatomy, they say the feather is a horny outgrowth of skin peculiar to the bird, but similar in structure and origin to the scales of fish and reptiles. So not only is the structure similar, but the way they develop, their origin is also similar. Well, I was kind of puzzled by this. And some years ago, I had a lab tech working for me, and I happened to know she had a boa constrictor. And I says, the next time that rascal sheds its skin, bring it on in. I understand you can hardly tell reptile scales from feathers. And she was an evolutionist, by the way. Anyway, we put the skin under the scanning electron microscope. This is an instrument that can see in 3 dimensions at very high resolution. And that's the way the scale looks. In the middle, you see a scale, and it has a very hard keratin. And then around it, you see a softer skin. I mean, if it wouldn't be for that fact, the snake would be like a fishing pole, just stick right out there. But it bends because you have the soft skin between the hard plates. Anyway, that's a reptile scale. And after reading all these evolutionists, it said you really can't tell the difference, especially if you look microscopically. I thought, well, this is going to be interesting. So take a look at that scale, and then I'm going to show you a feather. And let's see if you can tell them apart. If you get confused, look in the upper left-hand corner. We tip you off as to which is which. But there's a scale, and here's a feather. What do you think? Should I go back and forth until you have a chance to? Why do people say they're similar when they're not? Possibly they're similar in some very subtle or very biological way that we're overlooking. So let's dig. Scales, first of all, are folds in the skin. It's like drapery plating. One scale is attached to the next scale. That's why when a snake sheds its skin, and it can shed its skin because it really has 2 layers, huh? There's one on top and one underneath, and the one underneath kind of just plugs right into the one on top. You can see where the scales poke right up inside of the upper scale layer. And so when a reptile sheds its skin, the skin comes off as a sheet. You cannot separate one scale from another. This is a folded epidermal sheet. A feather is a completely different structure. It's a follicular structure. It's like a hair. It grows out of a follicle. It's very similar to a hair follicle. And they grow individually. In fact, they don't just grow individually. On a bird, the primary feathers grow in matched pairs. Left and right, balanced, matched pairs. They are shed in matched pairs, and they grow in matched pairs. That way they don't become asymmetrical by chance and start going through the air like a boomerang. When you put that reptile skin under the scanning scope, by the way, that arrow is pointing towards the tip of the tail, and notice the scales are kind of on a bias with the long axis of the snake. Now that's the top surface. You can tip the skin over and look at the bottom side. Let's do that. And when you look at the bottom side, now the tail's to your upper left, head's to the lower right, and you can see it's like a bunch of little pockets. Those of you in the cheaper seats will crank up the power, and right where that little arrow's pointing, that's where a scale of the lower level fit right up in that pocket. So when the skin was shed, it lower scale just got pulled right out of the pockets of the upper scales. A feather just simply couldn't be more different. Feathers grow out of follicles. Follicles are tubes in the skin. Hair follicles and feather follicles, at least at first blush, look very similar. But no evolutionist believes that birds have anything to do with having evolved from mammals. And so they ignore the striking similarities between a feather and a hair. Now, I'm not suggesting birds did evolve from mammals, obviously. I think birds were created as birds on the fifth day of creation. Dinosaurs, by the way, were created on the sixth day, so you couldn't have gotten birds from dinosaurs. But that's a feather follicle. In the center, in the darker blue color, is a feather cylinder, and down at the bottom is a growth collar. All of the growth occurs at the bottom, the same as in a hair follicle. It all sort of looks like this. Have you seen a pin feather? When the feathers first start growing out of a bird, they look like a fat pin sticking out of the skin. There's a shell over the feather. The feather is arranged in a cylindrical structure. If you take the shell off and look at the image on the right, down at the bottom there's a little ring. And the shaft of the feather starts growing up from the ring on the back surface of the cylinder, as you see it here. And the little barbs you see starting in the front get longer and longer and move around the circle and join the shaft in the back as they grow up. And they end up taking these long helical roots from the shaft of the feather around to the front. And so the barbs are really kind of up against the shaft like this, only helical. So when the shell breaks off, then the barbs can swing out like this and then out. And the whole thing just opens up, coming out of the feather. Let's look at a feather in its mature form. We're going to cut off a little piece of the feather vein on the left, as you see it there, and we're going to magnify that little piece, and that, let's say, will include 3 barbs that make up the feather. So the feather vein is made up of barbs. And let's look at a typical bird. Obviously, this varies all over the place from a swan to a hummingbird, but average size bird. Average feather will have 1 shaft or rachis, right? Right down the middle. Each shaft or rachis will have 2 veins, 1 on each side. Everybody with me? Okay. On an average feather, each vein will have about 400 barbs, of which you're looking at 3, parts of 3. And each of the 400 barbs, on average, will have 800 little barbs called barbules. Those are the ones you see in the 3 barbs that have been removed. Half the barbules point towards the tip of the feather. They're called anterior barbels. And half point more towards the base. They're called the posterior barbels. And then each of the anterior barbels has about 20 little hooklets on it. And these little hooklets work like Velcro. Only I've looked at Velcro under the scanning electron microscope. It really looks like junk. It's amazing it even attaches. But you look at God's Velcro, which is what a feather is all about, and you'll see some beautiful, beautiful engineering. In fact, let's do that. We'll start with a feather at low magnification. You see the shaft, a vein on each side of the shaft. You see the barbs that make up the vein. And you can kind of see the barbules that are crisscrossing between each barb. Now let's crank up the power a bit so you can see it closer. You can see this was a dirty bird. There's some dirt on it. We have the barbs going across the field, left to right or right to left. And you can see the posterior barbels are on top, and the anterior barbels are underneath. They're crisscrossing between the barbs. You all with me on this so far? Okay, let's crank up the power a little more. We'll bring the shaft in to give you some orientation. So the rachis or shaft is kind of going across the lower right corner. You can see a couple of the barbs going straight up and out of the picture. And then the barbules that are on the top are the posterior barbels, and the barbules that are underneath are the anterior barbels. And it's the anterior barbels that have the hooks. But you can't see the hooks because the posterior barbels are covering them. Let's crank up the power some more. Now you just see 1 barb going across the field. You see posterior barbels coming in from the top. You see anterior barbels going towards the top of the field from that barb. And again, those anterior barbels have the hooks, but you can't see them because the posterior barbels are covering them. So let's remove the posterior barbels. Now you see the hooks. Now notice where the barbels do not cover one another, there are little plates that fill in so the air can't get through there. And those little plates in some birds are covered with a double interference layer, which is like an oil slick in water that refracts the light to produce the beautiful iridescent colors that you see, for example, in a peacock's tail. So there's a lot more going on here besides just a lot of gorgeous anatomy. Well, let's put it all together for you now. The anterior barbels are going from the lower left corner to the upper right corner. The posterior, they have the hooks, and the hooks are sticking up. You can see one right in the middle. The posterior barbels go the other direction, from the lower right-hand corner to the upper left. And if you look very carefully at the posterior barbels, you'll see there's a groove in them. It looks like a celery stick. And if you look at the lip on the groove, you will see that the radius of curvature of the lip precisely matches the hook. Oh, now this is nice. You know, if you get a screen door and you want to put a hook on it with an eye, it does no good to go out and get a great big old hook and a little bit of an eye, or a great big eye and a little bit of a hook. They have to be a match set. And the hook has to be the right length. Does no good for it to come out here. It needs to go there. So you see you have the hook, but the hook isn't hooked up. It's just kind of sticking in the air. So it's going to have a hinge to allow the hook to swing over. And when it swings over, it's got to land just right on the groove. And the lip of that groove has to be matching the radius of the hook. Well, let's take this whole thing and tip it over. We're looking at the other side of the feather now. Now the hooks are going down, not coming up. We'll crank up the power a little bit more. Can you see the hooks there? Oh, there's so much beautiful anatomy in here. If you look, the barbels have a little strut running down the back that strengthen them. It's sort of like a jumper strut on the mast of the sailboat, the spreaders. The hooks are disengaged at this point. Let's go to the highest power I'm going to bother to show you right here. And there you see 3 of the hooks that are engaged from the anterior barbel onto the grooves of the posterior barbel. Notice one of the hooks actually has a little guiding probe that guides the hook in. And if you look at the base of the last hook on the right up there, you can see the nature of the hinge. It's a strap hinge. That would be a hinge sort of like this. It allows the hook to move in this axis, but it doesn't allow it to move laterally because strap hinges don't go that way. They just go like this. As you can see, basically a reptile scale. You know, I'm wondering, could I even give this lecture in a biology department? I mean, would this be like teaching religion, showing how unrelated a feather and a scale is? Well, we're simply going to have to simplify this lecture. Things are getting way too complicated. So let's look at a hair. Bible tells us our hairs are all numbered. And if you made something this beautiful, you'd number it too. There is a human hair. In fact, that human hair, I actually plucked from my scalp several years ago when I could afford this kind of thing. The hair has cuticle on it. Looks like shingles on a roof. Little shingles. And those shingles are what makes a hair shirt itch. Wasn't that worth the whole trip down here to learn why a wool sweater itches? And if you take a hair and pluck it from your head, if you start from the tip of the hair and slide your fingers to the base, you'll feel a resistance, squeaky resistance. But if you start from the base and go to the tip, it's smooth. It's because one way you're bucking the cuticle, one way you're going with it. And the cuticle is very important because it wraps around some kind of stick-like cells, bundles of sticks inside the hair that are also dead. They're called cortical cells. And it keeps the hair together because if the cuticle were to break off, the cortical cells would split. You've got it, split ends. The most incredible function of the cuticle, however, is that if God would have given us monofilament nylon, the same diameter as hair, and put that in our head, we would really have a problem. You ever get a snag in monofilament line fishing? Break out the scissors and cut it off because you're never getting that snarl out. But with the cuticle, when one hair turns on the other, they kind of lock horns and you can comb it right out again. In fact, companies that make natural hair wigs have to get all the hairs facing the same way or you can't groom the hair. And then, finally, that cuticle forms the most incredible thing you could think of, and that's a hair lock. Did you know you have a hair lock? Oh, let's get into that. Let's start out from the beginning. And if you look very closely at her face, you can see that there are 3 types of hairs. Actually, you'll see 2 on her face. One would be the terminal hairs. These are the black hairs that you can see, the ones we usually think about. They're called terminal hairs. They're pigmented, they're various lengths, but usually plenty long enough to see. But do you realize that on her cheek, she has as many hairs per square inch as she does on her head? She has as many hairs per square inch on her nose as she does on her head. These little hairs are called vellus hairs. They're very tiny, they're colorless, and they have big oil glands, which is why that kind of skin is oily. Now, when you're born, you have every hair you are ever going to have. You never lose a hair or gain a hair. And the follicles are able to make different kinds of hair, just as feathers can be made different feathers during different times of the year for birds. We can make different kinds of hairs. A given follicle can make a terminal hair, or it can make a vellus hair. So when we think we're losing hair, we really aren't losing hair, just a terminal hair fell out once, and instead of another one growing in to take its place, a vellus hair grew in. And then to add insult to injury, you not only get a bald pate, but it gets to be a shiny pate because of those big oil glands. Actually, all the hairs of our body begin life as a lanugo hair. This is the place setter hair, and all follicles make a lanugo hair first. If you see a baby that's born premature, this happens to be my little grandson who was born at 7 months of development, and you can see the hair on his arms. Now, of course, evolutionists look at that and they say, well, there you go, that reflects our ape ancestry. You know, nobody calls my grandchild an ape. So the lanugo hair is the first hair to form. And it always forms in association with nerves because hairs are actually mechanical receptors attached to nerves. So when you wiggle a hair, you're jangling a nerve. And so even the little hairs, the little vellus hairs of our body, when the wind blows past our face, we get a laminar flow that moves those little hairs and we get that refreshing sense of breeze. Something gets close to your skin, you can just feel it kind of tingles. People say these little vellus hairs have no function. Well, they have a function, it's just not a very dramatic function, but our body's full of things that are nice but not necessary. Well, there's a hair growth cycle. Hair grows for a while, like toothpaste coming out of a tube. After growing for a while, it stops growing, goes into a resting phase, and then after a few weeks, it starts growing again. The important thing is the old hair gets kicked out of the follicle, and the new hair grows in behind it. If this weren't the case, hair would grow so long, stop, then grow so long and stop, then grow and stop. They just get longer and longer. Can you imagine the squirrels going through the trees dragging 15 feet of hair? But the important thing is when the old hair falls out, a new one will come in behind it. And it won't fall out until a new one starts growing in. So when you see hairs in the sink, I have good news and bad news. The good news is that hair wouldn't be in the sink if it weren't for the fact there's a new hair growing in behind it. What could be the bad news? The new one could be a vellus hair. So anyway, that's the way the follicles look in the skin. Anatomically, they're really complex. There's like 8 concentric rings that are forming the various layers of the hair and the follicle. And several years ago, I did some work where I took skin and sectioned hairs. You could actually say I split hairs. You can see the lower part of the hair has living cells in it, and the upper part of the hair is dead. And there is a locking mechanism. We'll look at it by magnifying that area right there. There's a locking mechanism right there, and on the surface of the hair, you can barely make it out, but there's like little shingles there. And then lining the hair follicle, the tube in which the hair grows, it has another set of shingle-like cells. Let's take a look at this. The lower half of the field is the hair. You're seeing its cut surface. It's been cut down its length. And you can see these tiles that are the cuticle cells of the hair. And then in the upper half of the field, the lighter tissue there, is the lining of the hair follicle. It has a cuticle facing the opposite direction. Get the picture? It's like a ratchet. The two are interlocked. They are so interlocked that if you try to pull a hair out of the follicle, you're going to take about 2/3 of the follicle with it. Try it. Pull it out of your head, you'll feel a knob on the end. So this is kind of a drawing of the way it looks. Lining the hair follicle is a cuticle. On the surface of the hair, an orange is a cuticle, and the two interlock. So you just can't pull the hair out. There is a cuticle lining the follicle tube, and it's that cuticle that locks the hair in the follicle. Let's crank up the power one more time. Now you're looking at the innermost layer of the hair follicle. If you look at right about in the center to the left there of the coating cells, you'll see what looks like a kind of a little donut. That's a red blood cell. Give you an idea of the magnification. And look how closely that cuticle locks. If you peel it away, you can see every little nook and cranny on the hair. Matches every little step on the lining lock. Now, this ought to be raising some questions for you. If there's a mailing tube around the hair, as it were, that's dead, that has a cuticle on it, that's locked into the cuticle of the hair, how does the hair grow? Because it's being locked in. I mean, it's great. You can grab an animal by the hair. You can't just pull it out. If it wouldn't be for this lock, you could just pull hair out like this. I don't think any mammal would survive without the lock, at least other than man. And so the lock locks the hair in the follicle. A person can be lifted off the ground by your hair if you weigh a little less than I do and have a little bit more hair. But the thing that's really incredible is the lock is like that red tube you're seeing in this picture. That lock actually holds that hair in there, and the only way that hair can grow is the lock has to move with the hair. You think, well, wait a minute, if the lock moves with the hair, then you're back to the original problem of the hair being able to be pulled right out of the follicle. But there are millions and millions of little buttons that button and unbutton, button and unbutton, button and unbutton, all along the lock sequentially to allow the hair to grow and still be under control. My friends, we're talking about a hair. This should be easy. Can evolutionists explain a hair? I don't know. Let's look and see. This is an evolutionist explaining the origin of hair. The elegant complexity of the hair follicle, which at first seems so difficult to explain in evolutionary terms, may be readily explained. It permits good rod to grow from a good hole. A mere refinement which could have occurred quite late in the phylogeny of hair. The model which has been presented is highly speculative, but this is inevitable due to the nature of the subject. And this is science. Well, let's conclude. We've talked about feathers and birds, and we've talked about hairs. Let's let the Lord talk about feathers and hairs in Matthew 10, verse 29 and 31. Are not 2 sparrows sold for a penny, yet not one of them will fall to the ground apart from the will of your Father, and even the very hairs of your head are all numbered. So don't be afraid, you're worth more than many sparrows. And Job 19, 26 to 27. And after my skin is destroyed, this I know, that in my flesh I will see God, whom I shall see for myself, and my eyes shall behold, and not another.